So within this unit, we've already talked about protein synthesis, starting with DNA, getting the gene from the DNA. So we started with DNA. One section of the DNA was called a gene. So there's bunches and bunches of genes within the DNA. We would get that code, and that code would be transcribed into mRNA. And eventually, we would take the information from the mRNA to make our final protein. And our proteins determined our traits. In this section, we're going to look at gene regulation. What do we do to check to make sure that our DNA and our mRNA contains the correct code? And what is it called when there's actually mistakes in that code? So for this section of notes, you're going to be on page 52. And again, we're talking about gene regulation. So with gene regulation, you want to think about the end products of our cells, okay? And what is the specific job for each cell? All of an organism's cells is going to have the same DNA, but the cells are going to differ based on the expression of the genes. What does the gene tell it to do? Gene regulation is the process which determines which genes will be expressed. So again, all of your cells have the same DNA. If you think back to our reading at the beginning of the section, all of our cells have the same DNA, but some genes are going to be turned on and some are turned off. For example, our red blood cells are going to have the gene to be able to carry oxygen to produce a protein that carries oxygen, but they're not going to have the gene to create color, like our eye color, like the pigment in our eyes. Different types of cells are going to express different genes, and this is going to lead to cell specialization. So we had our red blood cells. Red blood cells have the gene to be able to produce a protein that carries oxygen. We have a nerve cell called a neuron that produces a gene to make these myelin sheaths that allow them to actually carry a signal faster. Okay, your red blood cell won't have that gene turned on. And so really this is what helps lead to genes, or to cell specialization. Now sometimes mistakes can occur, and if a mistake occurs in the DNA, we actually call this a mutation. But mutations can occur during DNA replication or during protein synthesis. Replication, again, we're referring to DNA, mistakes in the DNA. Protein synthesis, we're referring to mistakes in the mRNA. Okay? Because those two molecules are what carries the code. Now, the result, if there's a mutation, the result could be that the necessary protein is not made, or it's defective. It doesn't work. It won't work. An example here of a disease that's caused by a mutation is actually sickle cell anemia. And what you see in this first picture is this is the original strand of DNA. This is the amino acid that's made from that mRNA, from that DNA and mRNA. But if there's a mistake, and right here you can see that a different letter was placed in there, then you end up making the wrong amino acids in yellow, the wrong amino acid. And when you make the wrong amino acid, you end up making the wrong protein. So when mutations occur, there's actually two main types of mutations. The first one is called a point mutation, and this occurs when a single base, a single nitrogen base, like an A, is replaced with a different base. So instead of having a T being paired up with it, here you can actually see a U is accidentally paired with it. And that's your mutation. That's your point mutation. And this may result in the change of a single amino acid within the protein. So again, here's our original strand of DNA. This is the amino acid chain that we're supposed to make. But when we have this point mutation, we end up making the wrong amino acid chain. And when we make the wrong amino acid chain, we make the wrong protein. So sickle cell anemia is caused because of this. And if you would take a highlighter or your pen and kind of put a box or highlight this T with this G. This is your mutation. This is how it's supposed to be. This is normal. And this is our mutation. And write that in there as well. Okay? Now we do have one more type of mutation. The second type of mutation is called a frame shift mutation. And a frame shift mutation, and this is going to occur when a single base is added, we give you an extra nitrogen base, or we delete a nitrogen base. When this happens, the earlier it happens in the gene sequence, the base is added or deleted, the more amino acids will be changed. And what does that mean? So if you look at what was normal, if I end up adding an extra one, or actually in this case we're deleting one, notice that I have AUG, A, this is what was normal, but here I have AUGCCC. I actually deleted that A there. So if I delete that A, how do we read things? We read things in sets of 3s. So if I'm supposed to read it in sets of 3s, now it moves everything over, and I end up missing an amino acid and starting to make the wrong amino acids right after. So a disease that happens in humans that's caused by a frame shift mutation is Huntington's disease. And if you notice in this picture, again, I want you to have your highlighter or pen, put a box or highlight this C, okay? And here we actually deleted the C, so let's kind of put an X through it. And once you delete the C, well now the A that was here has moved up, and everything else behind it has started to move up. And when that happens, we actually end up making the wrong amino acids, so the wrong, completely wrong protein. And that'll definitely cause a problem. Okay. So here's just an overview of that picture again, showing you our point mutation and showing you our frame shift mutation. Notice in the point mutation, we only have one wrong amino acid. But in the frame shift mutation, possibly everything after that deletion or addition, adding an extra or taking away one of those bases, we have wrong amino acids produced. Okay? So today for your summary, and this is actually going to be extra credit, please try it because this can actually replace one of your missing homework grades. But for number 1, first thing I want you to do is create a sequence of DNA that is 6 nitrogen bases long. So number 1, create a sequence of DNA with 6 nitrogen bases. So make sure you remember what bases are in DNA. Then number 2, I want you to create the same sequence with a mutation. Okay, create a mutation of your own. Don't copy the ones from your notes, but use that as a guide. And the third thing is I want you to tell me what type of mutation did you create. Make sure I do want you to write everything in sets of 3s, so make sure you keep that in mind as well. And that's all for this unit.